Hi everyone, good afternoon and welcome to NRGene's webinar series. My name is Yana and I'll be your moderator for today. Today we'll be discussing an innovative mapping and trait marker service called TraitMAGIC. We will save time for questions at the end of the webinar and we encourage you to type in your questions during the presentation using the questions tab on the left of your screen. This webinar is also recorded and will be posted on our website in case you want to listen to it once again. We have Dr. Paul Chomet, NRGene Senior Technical Specialist, to guide us through this webinar. Paul has extensive knowledge in molecular breeding and biotechnology with over 25 years industry experience and a Ph.D. in genetics. He will share some insights about trait mapping and the development of markers and about our NRGene TraitMAGIC solution. We hope this webinar will be useful for commercial and academic breeders, molecular biologists, and geneticists who are looking for a complete solution to generate a genetic map traits, and start developing markers associated with such traits.
Thank you very much for the introductions, Yana. We're going to start today with a bit of background on NRGene, who we are and how we got started. I want to point out why researchers perform genetic mapping, and I'll give a bit of history on genetic maps. I'll introduce NRGene's service, which uses the latest technologies to both discover markers across a population and to perform efficient and sensitive QTL mapping to identify chromosome regions significantly associated to your measured phenotypes. I'll share a few past examples, then we'll finish up with some new offers from NRGene. Who is NRGene? NRGene's been established in Israel in 2010. Our initial range of products, including our genome assembly services, were released to the public around 2014. We now have a full set of tools for breeders that could reduce time and improve efficiency throughout the breeding process. Some of you probably know NRGene for our genome assemblies. We've delivered hundreds of assemblies for our customers, but we've also delivered unique analytics capabilities to some of the leading ag companies such as Bayer, Syngenta, and non-ag companies like Bridgestone Tires. NRGene has a number of products that fit together as a complete solution for different molecular breeding programs in plants and animals. Building up robust molecular breeding capabilities usually requires that physical map, a reference genome, and this is our de novo MAGIC product at the base of this graphic. Performing a detailed comparative analysis to understand diversity or genomic changes requires a comparison of genome sequences, for which we have the pan-genome comparison service called PanMagic. We also offer a TraitMAGIC service, which includes quantitative trait locus or QTL mapping and the delivery of trait-associated sequence markers. Essentially, this connects the dots between the molecular markers, such as SNPs, and observed traits. This is the capability that I'm going to focus on in today's webinar. Now, all of these capabilities are housed inside our GENOMAGIC system. This holds diversity information about a species as sequence-based haplotypes. The system can be customized and licensed for your breeding program. Associating a trait to a genetic and physical map is important in molecular breeding. It's also important for understanding genome function as well as improving methods to perform molecular breeding. Many marker-aided breeding programs are based on these trait-associated markers, and this can increase the breeding efficiency of a program. Now, on the left, here we see the association of markers to southern leaf blight resistance in maize. The identification of polymorphisms associated with favorable traits is essential in using genome modification tools such as genome editing and transgenic manipulation. And in addition to the trait marker discovery on the left, identification of important chromosome regions can be combined with the latest methods of molecular breeding, such as genome selection, to improve plant selection models. Now, in all cases, the use of trait association to genomic locations is essential. Now, a bit about history here. Historically, genetic maps were first developed using single traits that segregate in populations. Of course, it started with Mendel's laws. Mendel described the laws of inheritance and independent segregation, where there's a 50% chance of inheriting one of two alleles from the parent. So no map created here. It was all independent segregation. It wasn't until the early 1900s that Thomas Hunt Morgan's lab studied multiple traits that didn't segregate independently, but rather co-segregated in the offspring of fruit flies. This led to the statistical description of this association as a percent of recombination, and it describes genetic distance, with units designated as Morgans in appreciation for Morgan's discoveries. A few decades later, McClintock and her first grad student, Harriet Crichton, proved that meiotic recombination of markers was actually through the physical exchange of homologous chromosomes. But it wasn't until the hereditary material was discovered by Hershey and Chase in 52, and the structure of this DNA molecule discovered in 53 by Watson, Crick, and Franklin, that it was understood what the molecule was that was being exchanged on the chromosomes that McClintock was looking at. Back then, and even today, we can map traits using phenotypic variants as markers, but methods to detect DNA variation, which would be independent of phenotype, became available as markers. This was ushered in by the Southern blot technique, and the first restriction fragment length polymorphism maps were produced in the mid-1980s. And here you see one of the first maps of maize using these RFLP markers. Now today, we have efficient sequence capabilities, and therefore ideal maps contain sequence-based markers, usually at a very high density, and they're anchored and positioned on a physical map. Many technologies allow for assays at predetermined positions relative to a single genome. These preset markers are usually based on single nucleotide polymorphisms, or SNPs, and oftentimes utilize a reference genome, which is depicted on the left. Therefore, there is a series of experiments that need to be done to identify the appropriate positions of these markers, as well as the allele frequency of the SNPs in your germplasm. These data allow for manufacture of a DNA chip or other fixed assay methods to be developed. Since the fixed markers are usually used across a variety of germplasm, these markers are selected to increase the ability to detect polymorphisms, to use it in mapping. Therefore, one balances the ability to assay with common polymorphic markers versus the ability to detect more rare or unique alleles. Now on the right, the latest methodologies based on sequence technology de novo discovers the near full sequence of each parent of a population, and these sequences are arranged to represent the actual parent chromosomes using a reference genome. In this next gen mapping capability, we can de novo discover markers for each parent. This technology gives an abundance of markers, which allows for several very important capabilities. One, I said, is the de novo discovery of parent markers. Gives the capability to identify key sequences associated with both common and rare haplotypes. The high density of markers allows for higher accuracy in defining the recombination breakpoints in the population. And the large number of markers can reduce the complexity of association statistics by transforming that high number of markers into a lower number of parental haplotype blocks. With such high density maps, one of the key issues has been how to take advantage of this dense marker information, but still optimize association statistics for detection sensitivity and reducing false positive detection of a QTL. Now, NRGene has utilized this next-gen whole genome sequencing to first discover these parental sequences, which are unique between the parent lines, and order those sequences on a physical reference map, which we can produce as well. Parental sequencing and analyses discovers hundreds of thousands of sequences that distinguish one haplotype line from another. Progeny are then used to build such markers into phased haplotype blocks in the population. The blocks can then be used as polymorphic chromosome segments for trait association. This reduction of millions of markers, in some cases, to a full description of recombinant segments allows for robust and efficient statistical methods to detect that QTL with high sensitivity. Now, separate genetic maps are developed for each haploid genome when we use a heterozygous parent in a mapping population. This makes the system really well suited for outcross species. Depending on the sequence depth, this method can improve the detection accuracy of the recombination breakpoints, improving the precision in QTL mapping. On the left, we see a set of fixed SNP markers across a reference genome. Let's say we're using a 50,000 or 50K chip. The genotype of a single progeny is shown below, where the markers detect the parents A, B, or are monomorphic, or not distinguishing between the parents. So A is light blue, B is the darker blue, and monomorphic is represented as gray here. As you can see depicted in gray in the progeny chromosome, many times markers are monomorphic between any two parents, and there's regions of uncertainty of the parentage. And those fall into that region of uncertainty marked in the graphic. On the right, using sequence-based haplotype markers, which we discover and map to the position according to the map, we discover a very high density of unique parental markers and put them on the genetic map as well as infer them from the reference genome. Depending on the sequence coverage, the region of recombination can be refined, and we usually use more than 0.1x coverage of the genome, and therefore we can identify breakpoint precision of less than 10 kilobases. As I just described, the unique haplotype parental markers, unique for each haploid genome, are utilized and genetically guided by the progeny for their arrangement. The system then reduces the complexity by taking hundreds of thousands of discovered unphased markers down to phased haplotype blocks, which are handled as genetically and physically defined chromosome regions, with a single genotype call per haplotype block. The parental sequences are used to impute the haplotype blocks. Trait association is performed in the QTL mapping using a simple interval mapping. Since the complexity has been reduced and this method is robust for single haplotype blocks. Accounting for the multiple QTL is also used to reduce the phenotypic variance caused by link QTLs and gives better estimates of QTL effects. In the case shown, you can see that we handle the mapping of traits for each parent separately. This allows the more sensitive detection of independent QTL from each parent and the ability to see if each parent accounts for variation of the trait at each locus. There are a number of advantages to this service. This includes the genotyping is performed with high-density de novo marker discovery in your population. The QTL analysis is performed on the phased haplotype blocks, which helps in reducing the false positive and the false negative QTLs discovered. The service is really ideal for heterozygous parents. The QTL discovery is performed for each parental genome. The other nice thing is the service is all-inclusive. We just require a reference assembly of the species, or actually we can assemble your genome using de novo magic. Like any mapping population, it does require a population of progeny, Approximately 200 lines in a standard mapping population is good. And like any method, it requires high-quality phenotypic DNA. As a customer, you can contact us through the website or email as listed. You provide us with the parent and biparental population DNA, or tissue or sequence data appropriately fitting our analyses, and we'll return the high-resolution physical map as a VCF file and a bed graph of the QTL analyses along with a summary. The entire service is often less than 8 months from start to finish, and if a reference genome is needed, we can perform this service first and use the map to build the scaffold into pseudochromosomes. Now, let me show you a little bit of the results that we see. In this example, we've utilized a population from the Maze Nested Association mapping population. The very low coverage of the public GBS dataset was used here to impute to the population parents, B73 and CML247. You can visualize the recombinant inbred lines as rows in this graphic, with blue representing B73 and pink representing CML247. The columns represent the 10 chromosomes of Maze. Now, Bryan et al. Published southern leaf blight resistance phenotype data on this population. And we utilize this data to QTL map resistance in the population, and above the genetic data in this graphic is a graphic or a graph showing the LOD score explaining the variance for this trait. As you can see, and as the published paper found, there's significant QTL on chromosome 3 as well as other locations. Now, if we look underneath this QTL, we can do that with the files that are delivered, the VCF files. It identifies an abundance of unique sequences that define the region and the CML resistance haplotype and can be utilized to help design markers for this QTL. This VCF file, along with QTL positions, are delivered as part of the service. As many researchers do, we look underneath the QTL for candidate genes. This is normally done for a standard reference genome such as B73 for maize, but the B73 does not carry the resistance allele. Now, Bryan et al. Identified an NBS LRR type gene, a resistance gene, as a candidate causal gene. But what does this gene structure look like in the resistance locus? In this case, we've previously assembled the parental line CML247 and could look at the polymorphism in this figure from NRGene's IGV genome viewer. We can zoom in on the NBS LRR candidate gene, shown here, and detect all the polymorphisms between the putative resistance locus in CML247 and B73. Now, as indicated by the coloring, there are significant changes to this locus. And in this case, we can see in gray that the promoter and exon exhibit a deletion as compared to B73. This allows for very specific hypothesis testing to occur to determine if this is the causal gene for the resistance. In a paper published last year, NRGene utilized the TraitMAGIC system and genotyped a recombinant inbred line of melon using sequence-based haplotype markers. There were a total of 58,000 markers discovered. These were phased, and as I described, these were used to identify a much fewer number of haplotype bins, 3,600 haplotype bins, which captured all the recombination events in the population. These were then used to identify 241 QTLs, describing 129 fruit traits. For more information about this, please see the paper by Galpaz listed in the slide in 2018. Now, I want to just sum up what I've talked about, that we have an efficient and all-inclusive QTL mapping service. This service discovers and maps high-density sequence markers in your population. It's extremely sensitive and accurate for QTL discovery, and it performs well in outcrossing species. Now I'd like to switch over to just also tell you about some new offers from NRGene. We're currently running a few consortium for a number of crops like canola, cotton, pepper, brassica, sunflower, and tomato. If you decide to join one of these consortiums, you'll receive all the consortia assembled genomes, and you're going to benefit also from our PanMagic service with a new pan-genome visualization tool. All of this will be with no additional cost, and we also have reduced prices of the de novo magic services. Additionally, we came up with new types of library recipes for genome assemblies for homozygous triticity that allows us to offer a very competitive pricing on genome assemblies. I want to invite you to reach out to NRGene or to an NRGene representative to ask for more information.
Thank you, Paul. I believe we have some questions sent to us from the audience. So here goes the first one. How many progeny should I use for my biparental trait mapping population?
Thanks, Yana, and thanks everyone for sending in questions. We're going to get to every question. If we don't answer it here, we'll certainly get to answer it and send it back by email as fast as possible. So for this first question, how many progeny? Well, this is somewhat dependent on population structure and recombination events in the population. But in general, if you have an F2 population, a back cross, or even an F1 pseudo-test cross, we like to see at least about 200 progeny. Of course, more recombination and therefore more progeny will likely give improved resolution of a QTL, but 200 is about right.
So to the next question, it will be, will the markers delivered be applicable to other varieties?
Good question. So where is this in trait marker discovery for populations other than what you're utilizing here? The identified marker sequences, of course, in our TraitMAGIC are polymorphic between the parents of the mapping population. That's a really important starting point, and it identifies the parental haplotypes associated with the trait. If the haplotype that carries the trait will be used to introduce into new germplasm, the markers are going to need to be tested across that breeding germplasm, and then select the appropriate markers that uniquely mark the associated haplotype. And this is how trait markers get built and turned into a marker system.
So in our next question, and unfortunately it will be our last, is how much DNA do you require for the parents and for the progeny sequencing?
We require about a 0.5 to 1 microgram of DNA per sample, depending on the size of the genome. Now, we have a specification sheet that outlines this, the quality, the concentration, and the quantity of DNA, which we'll be happy to send to you if you email us and give us your information, and then we'll send you back that spec sheet.
I'm sorry we couldn't answer all the questions, but please note that we will answer them in writing after the webinar. Thank you so much, Paul, for taking time and sharing from your expertise. If you missed any part of this webinar or wish to listen to it again, you'll be able to find it on our website. Thank you for joining us today and enjoy the rest of your day.